Hello, this is CJ, and this is Knowledge Catalog, and in this video, we are going to talk about a simplified version of the central dogma, which is a key concept in molecular biology. So let's start. The first step is replication. In replication, there is going to be a net yield of two identical copies of the parent DNA, and this ensures the transmission of information to the daughter cells with high fidelity. It is done with the initiation of topoisomerase, which is cutting a strand to release over one or under one spot. The helicases then separate the strands of the DNA, allowing the RNA primers to initiate the replication process by the DNA polymerase, and once it is done, the replication is through as well. So the point of replication is to have another copy of the parent DNA. So the parent DNA is going to yield two daughter DNA. So there will be two copies of the parent DNA. The next step is transcription. The DNA that was produced through replication is going to be used in transcription, or the DNAs, the daughter DNAs. So in transcription, it starts with the unwinding of one of the daughter DNA strands through the DNA helicase. And then the mRNA, the messenger RNA, which is ribonucleic acid, that contains nucleoside triphosphates, will provide complementary nucleotides to the template strand. The other strand of the DNA is called the coding strand. So yung tinatranscribe in this process is actually the template strand. Yung hindi, it is called the coding strand. So from the DNA, the raw material DNA, transcription is going to have to yield an RNA, which is the mRNA. So that is the product of transcription. Now, the same mRNA that was produced through transcription is going to be the raw material for translation, which is the last step of the central dogma. That is, if we're referring to the usual flow of activities. So translation involves the protein synthetic processes where the mRNA, the messenger RNA, goes through a ribosomal pair where it is read by a transfer RNA molecule that brings complementary nucleotide codes. The process of producing a polypeptide chain starts with AUG, that is an adenine, a uracil, and a guanine, the start codon, which is coding for methionine. And then it's going to count by 3. Bali yung coding niya is by 3. And then it's going to start reading by recognizing age. And then it's going to stop with UGA, UAA, and UAG, which are the stop codons. Again, in translation is the actual protein synthetic process. The end product in translation is a protein. And for the coding to begin, it requires the presence of AUG, which codes for methionine. And once it is identified, you start counting by 3. And then by counting by 3, you will be able to code for alanine, phenylalanine, and the others. And then once you see any of the 3, UGA, UAA, and UAG, you need to stop, it's going to stop coding because those 3 are stop codons. So I hope that we were successful in simplifying the central dogma. It is one of the most important things that we need to keep in mind if we are to understand some other bigger processes in biology. Again, this is CJ, and this is another video for the Knowledge Catalog Education Solutions.